The overall goal of the following experiment is to study the motions of protein molecules and protein-coated beads with the ends of depolymerizing microtubules. This is achieved by assembling a reusable flow chamber with a clean, sialinized cover slip. Next, microtubule seeds are prepared, attached to the cover slip, and used to nucleate microtubules, which are then stabilized temporarily with photodestructible caps. Then, GFP-labeled proteins or protein-coated beads are introduced to the flow chamber and bind to segmented microtubules. Microtubule depolymerization is then triggered by removing the caps by photoablation. Images are analyzed with chymographs to determine whether the labeled proteins exhibit tip-tracking behavior, which is movement with the ends of depolymerizing microtubules.
The main advantage of this technique is that it allows triggering microtubule depolymerization with high temporal and spatial resolution. The visual demonstration of this method is critical. The protocol begins with flow chamber preparation and assembly.
Using ultrasonic milling, modify regular microscope slides to have 2 grooves. Make the grooves 14 mm apart, give or take 1 mm, which is optimal for chambers assembled using square 22 mm cover slips. Next, place a 100 mm long polyethylene tube in each groove in the slide, leaving about 5 mm overhangs at the inner ends of the grooves. Fix the tubes inside the grooves with cyanoacrylate glue, embedding the tubes completely inside the grooves. Carefully